And everybody, I hope everybody's doing well. At the end of this week, we are about to go into the Labor Day weekend, but I wanted to do this presentation because I was really passionate about it. It's going to really allude into the conversation that the gut microbiome is having and how I can use the data from BiomeFx to be able to eavesdrop on these conversations. And better yet, what if we could change the narrative of what's being said? So I'm going to jump right in and talk about quorum sensing. I'm very passionate about quorum sensing. It's what I did my thesis on. And quorum sensing is how bacteria communicate with each other. It is actually their molecular signaling to each other in order to change their genetic output to shape an ecosystem. So in gram-negative bacteria, we have AHLs, or N-acetyl homoserine lactones, and those are how gram-negative bacteria will talk to each other. And in gram-negative bacteria, those AHLs just diffuse passively through their membranes. But in gram-positive bacteria, they use auto-inducing peptides. And there's a whole, energy-dependent uptake through their membranes that they utilize. But the coolest part about this is that they need density. They need density of their family in order to have this signaling take effect. So if there isn't a lot of them, then they can't quorum sense their entire ecosystem. They can't make a large impact because not everybody is getting these signaling outputs. And how that affects us is based on the density of certain family or phyla, we can shape an ecosystem. And really by understanding what molecules are being produced by these bacteria, we can better understand the conversations that are happening in this ecosystem. So in the last 91 test kits that I've done, 50% of them are Bilophila wadsworthia positive. So they have this pathogen, Bilophila wadsworthia, and I'm seeing it really pervasively through all of the BiomeFx test kits that we're getting back. And when I get this data, I'm seeing and this is one of the things that I love about BiomeFx is that it doesn't just tell me that it's there, it tells me its abundance, which in quorum sensing is super important. Remember, we're talking about density. And it says, so obviously this person had Pseudorella wadsworthiensis as well, but just looking at Bilophila wadsworthia, as long as it's in that 0.0 to 0.126%, then Bilophilia is in its cage and it's not behaving pathogenically. More importantly, its signaling molecules aren't making it to anybody to have any sort of huge effect on the human. But in this person's sample abundance was 0.555%. So much higher than it being in the cage. Bilophilia at this point has come out of its cage and it's kind of wreaking havoc on the ecosystem. And now it's able to send out its quorum sensing molecules or its AHLs. And what happens when the other neighboring Bilophilia takes up this AHL is it goes down into its genome and it becomes a transcription factor. And it transcribes the genes that upregulate virulence and many other, physiological changes that are wanted by the ecosystem in order to behave pathogenically. So for Bilophila wadsworthia, we see that it upregulates the genes for hydrogen sulfide production. And it also induces this low-grade inflammation that was suspected, but really specifically, we're able to see that it does induce amyloid A and the upregulation of interleukin 6. In this study in 2017, they took 2 germ-free mice, which they do in microbiome testing is we basically wipe out the genome of these mice. And then they just specifically inoculated one of the mice with what was comparative to a healthy, balanced ecosystem. And then they inoculated the other germ-free mice with just Bilophila wadsworthia. And they were able to see that, yes, Bilophila wadsworthia does have this impact in upregulating inducing interleukin 6. So the way that this communication, now that Bilophilia is dense, the way that this disrupts the microbiome, and really the eavesdropping that we can tell through the BiomeFx testing, is Bilophilia is going to start generating high levels of H2S, and it really promotes the quorum sensing signals, or the transcription of genes that are, causing virulence in the ecosystem. But also it suppresses commensal signaling. So the good bacteria are not able to have their conversations because Bilophila wadsworthia's quorum sensing signaling is directly interfering and suppressing that communication. And it actively pushes the ecosystem into an inflammatory and dysbiotic state. So if you look here at the bottom of the page, of the screen right here, BiomeFx in this same sample was able to tell me that, yes, this person with that 0.555% abundant Bilophila wadsworthia is also creating H2S in the 97th percentile. So I didn't have to just assume that the density of the population is upregulating quorum sensing genes. I was able to specifically eavesdrop on what the conversation is. And we know that hydrogen sulfide acts as a signaling molecule as well. So yeah, they are having some alterations in their gut microbiome. It's being reshaped. It's increasing gut permeability. There's inflammation. The transcriptomics of this landscape are to support and to encourage bilophilia. And we don't want that, because we need a lot of other of our keystones and our commensals to be able to have this environment the way that they want it. And it's reducing the abundance of those commensals, and really the quorum sensing signaling is leading in the direction of inflammation. So in the clinical implications, the way that you're seeing this from a clinical perspective is this person now has altered bile metabolism, because biophilia loving, this bile-loving microorganism is interfering with bile acid metabolism. And we need bile acids to be converted to secondary bile acids by our gut microbiome, the ones who are intended to convert them, and not interfered by bilophilia to make its own taurine conjugated bile acids, so that we can have healthy metabolism. We see that this upregulation of H2S and ethanol affects the liver and causes fatty liver disease, but it's also causing like fibrosis of the liver. There's increased gut permeability because there's a physical depletion in short chain fatty acids, which, remember, the importance of short chain fatty acids is abundant, and we could literally do hours and hours long podcasts on the importance of short chain fatty acids. But really, if we just want to talk about the hallmark short chain fatty acid, butyrate, the objective of butyrate is to be the energy source for the colonocytes in the colon. Remember, the energy of all of our cells comes from oxygen and sugar, and we need carbohydrates, we need sugar, we need water. Okay, and it's able to have respiration, and we're able to develop ATP from there, which is our cell's natural energy source. But in the colon, it is an anaerobic ecosystem. There is no oxygen, and so these cells have evolved to utilize butyrate as their energy source. So when you deplete short chain fatty acids, you're physically removing the energy source from the cells, and you're really wreaking havoc on the integrity of this lining. Inosine is also depleted, which is an important nucleoside in RNA synthesis, and the liver becomes inflamed, and there's that bile acid homeostasis that's disrupted, mostly because the FXR receptor has this altered signaling. And these FXR receptors need bile acids in order to be a ligand to promote healthy glucose metabolism, increase insulin sensitivity, but also have normal lipid metabolism. And so the metabolism is everywhere. And in that same study in 2017, they took those germ-free mice that they inoculated with what was reflective of a healthy ecosystem, and then the other one, which was the Bilophila wadsworthia, and they weighed the spleen and the liver after, these mice so lovingly offered themselves to science, and they found hepato and splenomegaly in both of these mice because of the fibrosis that's happening from this inflammatory process. So this isn't just dysbiosis. This isn't just, an imbalance in the ecosystem. Sometimes I look at Dr. Noel and I'm like, does this person have fatty liver? Because if I've never met them before and I look at their data and I eavesdrop on this conversation, I'm like, if they don't now, they will, at some point. And it's because these mechanisms are very well understood. So have no fear. Our hero is here. I give these people HU58, and it's one of my favorite Microbiome Labs products. And it's the most well-studied gram-positive bacteria, Bacillus subtilis, specifically the strain HU58. So my hero is a powerful communicator of eubiosis. He's going to go in there, and instead of producing quorum sensing signaling molecules, he's going to produce quorum quenching signaling molecules, physically going in there and interfering with the virulent signaling that's happening in, for example, in this example, but there's hundreds of examples with Bilophilia. And it's going to interfere with Bilophilia making its biofilm, which is another virulence transcription factor that happens. And in vivo studies actually showed that Bacillus subtilis, why do they all have to have very similar names? In vivo studies showed that Bacillus subtilis actually makes pathogen-specific peptides. So it's not like a one-size-fits-all. This guy has evolved to be able to say, oh, you Bilophilia, you and I go back, and I have a peptide for you that I know is going to targetedly diminish your abundance in this ecosystem. And it really is just really good at outcompeting pathogens and making them more vulnerable to the host's defenses or immune cells. And the way that it does this is we talk a lot about gram-negative bacteria and the lipopolysaccharides that are on their membrane. So I'm going to kind of zoom in just a second here. So this is a gram-negative bacterial cell wall, and these lipopolysaccharides that make up this outer membrane, we know how inflammatory they are. But let's talk about gram-positive cell walls. And Bacillus subtilis is a gram-positive bacteria, and it has these wall-tocaic acids and these lipotocaic acids. And what it does is it actually physically modulates the immune system by interacting with those pattern recognition receptors on the surface of our immune cells, right? We talk about those receptors that are mediated in our immune cells, and it says, hey, we're good. I'm here. You can chill. We're going to recalibrate the ecosystem. I know Bilophilia has been running amok around here, but it uses its wall-tocaic acids to interact with those pattern recognition receptors on our immune cells to say, we're good, you can calm down, and physically reduces the inflammation that way. And it downregulates pro-inflammatory cytokines. So, besides the lipotocaic acids or the wall-tocaic acids, it also produces lipopeptides that disrupts the membranes of pathogens. It's this surfactant that kills the pathogen, but also influences the host's immune response to create more balance. And then it also has these extracellular vesicles, these EVs, that are just antimicrobial peptides that go in, and these are like those targeted pathogen peptides that I was talking about a second ago. But also remember, gut permeability, like the intestinal permeability, is high. And so Bacillus subtilis is actually making the components of the cell of our intestinal lumen to help with repair. It's making claudins, it's making zonula occludins, it's making the proteins so that our, large intestine can repair its barrier. And then it also has this dipeptide antimicrobial called bacillicin that has this like smart release of targeted pathogen remediation so that it's not this like widespread dysbiosis, right? It's like, oh, okay, I found you. We're gonna take you out. Oh, I found some abundance over here. Here's some bacillicin, take that. And that way it's not causing more imbalance like we would see in an antibiotic. So then there's a battle, right? We put in HU58. There's this abundance of Bacillus of Bilophila wadsworthia, like we saw in the testing. And so now these antimicrobials are battling these organisms. And what happens is that during this kill off or this die off, we've all heard about the Herx reaction. It's going to release these extracellular, products, this debris, these toxins, and the body responds to them, right? It doesn't like all of that. So the byproducts temporarily will overwhelm the system, and it causes these symptoms of these toxins being high. So gastrointestinal, discomfort, you got loose stools, diarrhea, fatigue, headaches, sleep disturbances. This is another one that I hear about a lot. And as a provider, you're like, no, because that help is on the way, and that they just need to brave through this period of time because the biome didn't become imbalanced overnight, and it's not going to become rebalanced overnight, and there's going to be a battle. We have an overgrowth of a live organism, and now we're taking him out. And so what I love about Microbiome Labs is they offer this product, because every Batman has his Robin. And that's Mega IgG 2000, which is going to go in and physically bind up this cellular debris, this LPS, and neutralize these toxins. So it is the perfect wingman to HU58. It helps, the gut barrier by, reducing immune activation. It detoxifies. It's 2,000 milligrams, which is such an efficacious dose for this battle. I talk about HU58 like he's the Navy SEAL, and this guy's going to go, he's at the same level as the Navy SEAL in being able to combat and bind up and work synergistically with HU58. So, who goes first? Do you give HU58 first, or do you give Mega IgG first, or do you do them both at the same time? And what I've learned is low and slow is the best. Your clients, your patients, and so you have to really feel out what's going on. Are they already having constipation? Are they having loose stools? But what I've learned is no matter how they're presenting, I usually prefer and they tolerate Mega IgG 2000 first. So I'll do a week of Mega IgG 2000, and then on the second week, after Mega IgG is already in there, kind of suppressing some of that, overactivation of the immune system and that inflammatory response, then you can go in with HU58, and Mega IgG is already sitting there. It's already been there. It's already waiting. It's already created the infrastructure for HU58 to go in and battle. And remember, HU58 is a taper. It's a taper up. So the first week that you're on HU58, you take 1 capsule a day. And then the second week, you up that to 2. But every client, every patient is different. They're clients to me because I'm a scientist, and they're patients to you. So that's usually how I've been successful, is introducing Mega IgG before HU58. So, I just want to wrap up my little monologue by saying that BiomeFx is a sophisticated tool to eavesdrop on the dialogue of your gut microbiome. We know that these communities are producing signaling molecules to, mediate their genomes and create ecosystems that are beneficial for them. So if you would like, you can, give us a call, and we'd love to offer you a discount to our viewers of $50 off a stool kit if you just use the phrase mastermind. This is and that's it. These are my references. If anyone wants them, just reach out and it's okay. All right. That's all. If you have any questions, please let me know. I would love to do a BiomeFx test kit on you, or talk about how we might be able to create some calm in what's likely a very imbalanced ecosystem.
Well, I have a question.
Sure. You're allowed.
You did such a great job that the whole time I like, oh, she's answering my question. I wrote a few of them down and then you answered them.
Okay, good.
When you did this talk in front of clinicians.
Yes.
And I know they had lots of questions. I didn't get to hear all of them, but there was one that to me really stood out.
Sure.
And it's one of those questions, honestly, I get this every day with patients, is about testing. You know, you use the microbiome labs testing, and there's a lot of people out there that come in, well, I already have my testing from this lab or that lab. Yeah. And it's not to put any of those down, but there's a reason why you use the microbiome labs testing. You're not paid by them to say that.
I'm not.
It is
but if you would like. No.
Yeah. Send a check over here.
Let us know. No, I'm not.
But can you just kind of briefly, I know you've talked on this before on other talks and other blogs and things like that, but
for sure,
what is the main thing that you want to say about microbiome labs, the test that they have as it compares to others?
Sure. So again, this is no flack to any other company that's characterizing the gut microbiome. We are in the genomic era. And, you know, we sequenced the first human genome like 25 years ago. That's not that long ago. And so our technique for being able to characterize DNA has come a long way, but it's also changed rapidly. And a lot of the tests that are on the market right now utilize what's called qPCR, which is a tried and true methodology that we need, especially when we're looking at very specific microbes, or, you know, we're trying to sequence very, you know, known genes, or we kind of have an idea about them. But it's not as wide net. It's not as much of a wide net cast as whole genome sequencing is. So let me put it like this. During COVID, we were using PCR or qPCR to be able to say, yes, you have COVID or not. And that's because we were looking for COVID. It's not like they swabbed your nose and they were like, you have COVID, you have the flu, you have some weird streptococcus. They weren't doing that because they can't do that unless they're looking for it, which creates a bias. So qPCR says, I want you to look for streptococcus, bifidobacterium. I mean, there's trillions of them. So I want you to look for these 10. And then the test comes back and is like, yeah, those 10 are there. What whole genome sequencing does is it says, here's the sample of stool. I want you to sequence everything that's in this stool. And then I want you to tell me what's there. It's very specific. It's very nuanced. It's the most up-to-date technology as it comes to genomic characterization. And it's now commercially available where it wasn't before, because we were still figuring out how to do it at such a high speed and with a good turnaround. But because the genomes of our bacteria and our yeast and our viruses are so small, we can sequence the trillions of them in 4 weeks through microbiome labs, BiomeFx, which is just incredibly impressive. And so I've been able to find genus species that wouldn't normally be in a microbiome and say, oh, you actually have an oral microbe that is down there that's causing some harm. I mean, I found all kinds of crazy stuff, and it's because the test is telling me who's there. I'm not asking the test who is there. And when we're talking about relationships of balance, being able to decipher the trillions of microorganisms that are in this ecosystem, being able to look at the ratios, which tell us is there biosis, eubiosis, or dysbiosis, becomes far more accurate. When we can look at something from the phyla taxonomically down, I mean, we can say some things with more confidence, but we're also able to sequence the metabolites. And so in other testing, it's like, yeah, this guy is there, but I don't know how he's behaving. Now I can tell you who's there. Is his mama there? Is his cousins there? And are they producing the metabolites that we need?
Outstanding.
Thanks.
So to wrap it up,
okay,
it's all about relationships.
It's all about relationships.
Yeah.
Yep.
Even for those little ones.
Yeah.
You know,
including, don't leave them out.
Yeah.
Don't forget about them.
All right. Thank you so much.
Absolutely. Take care, guys.